There have been several questions about the fed and fasting state, so I thought I'd take a moment to look at each one of those, and we'll start by taking a look at the fed state. So in the fed state, there is plenty of glucose around, so that is what we'll be using as our primary energy source. Recall that glucose will cause the secretion of insulin from the pancreatic beta cells, and so that is what's going to be the primary hormone in our fed state. This insulin is ultimately going to alter the activity of several key proteins that will result in the utilization of glucose as our energy source. It will also work on building up our energy reserves. And these are going to be the triglycerides and glycogen. And lastly, we can also, since we have sufficient glucose and energy around, do some synthesis of other biological molecules, such as cholesterol. We'll start by quickly reviewing how glucose causes the secretion of insulin from the pancreatic beta cells. So glucose is taken up into the pancreatic beta cells through the glucose transporter. Recall that this is a facilitated diffusion process, so it's only dependent on our concentration of glucose. Once inside the cell, the glucose can be converted into ATP by going through glycolysis, citric acid cycle, and oxidative phosphorylation. This increase in our concentration of ATP is going to cause a closing of our ATP-gated potassium channels, triggering a depolarization of the cell that opens our voltage-gated calcium channels located at the bottom of the slide. This increase in calcium can cause additional releases of calcium from the endoplasmic reticulum, and the increase in intracellular calcium is also going to cause the insulin granules, so these are granules with our preformed insulin inside of them. It'll cause the granules to go to the plasma membrane, and then through exocytosis, we get the secretion of insulin. Remember that we had mentioned that these ATP-gated potassium channels are the targets of the sulfonylureas, which are drugs used in the treatment of diabetes. So these sulfonylureas can essentially cause the closing of the ATP-gated potassium channels, which will then again result in the secretion of insulin from the pancreatic beta cells. Following secretion, the insulin can cause the activation of phosphatase enzymes that regulate protein function, including those enzymes or proteins that are responsible for glucose uptake and glycolysis, so that's using our glucose as an energy source, as well as those enzymes involved in the formation of our energy reserves, so glycogen synthesis, fatty acid and triglyceride synthesis, as well as the synthesis of other biological molecules such as cholesterol. What I've written here are the proteins that we've talked about whose regulation you're responsible for knowing, and that is the acetyl-CoA carboxylase enzyme that's involved in fatty acid synthesis, our lipoprotein lipase, which is responsible for cleaving the triglycerides inside of the lipoproteins so that those fatty acids can be taken up into the adipose tissue, converted back into the triglycerides and stored for later, as well as HMG-CoA reductase, which is the key enzyme involved in cholesterol biosynthesis. If you're interested in the specific proteins whose activity is regulated by insulin, those are shown here. You can see that we have a number of the enzymes involved in glycolysis listed. We also have the several different enzymes involved in fatty acid synthesis and triacylglycerol synthesis. We've already mentioned ACC is one of the enzymes that we focused on. And then you also see some decreased expression with enzymes involved in gluconeogenesis and glucose release to the bloodstream. If we take a look at the net actions of insulin, those will be to increase the proteins that are going to be important for converting glucose into energy. Remember, glucose is our primary energy source in the fed state. We will also be working to build up our energy reserves. So we'll be increasing our synthesis of glycogen and triglycerides so that we can use those when glucose is no longer around. And we also have sufficient energy then to synthesize other molecules, including cholesterol. In addition to those activities that are decreased, we also see a decrease in other activities in response to insulin. Those will be to decrease glycogen breakdown and gluconeogenesis. We'll see a decrease in fatty acid mobilization from the adipose tissue, and its subsequent oxidation will also be decreasing ketogenesis. You notice all these have in common that these are our energy reserves. So if we are going to be synthesizing those energy reserves, we don't want to be simultaneously breaking them down. So that should make sense. You should also remember that we talked about how the fatty acid synthesis through the enzyme ACC produces malonyl-CoA, and it's the malonyl-CoA that prevents the fatty acylcarnitines from getting into the mitochondria, thereby preventing the fatty acid oxidation from occurring. So that's how we control or regulate the fatty acid synthesis and oxidation. Also remember that we are always going to link fatty acid oxidation and ketogenesis together. Since usually if we're using fatty acids as our energy source, we have to remember the brain can't utilize the fatty acids as an energy source. So therefore we'll be synthesizing our ketone bodies. So that takes care of the fed state. And let me know if you have any additional questions.